All right. Okay, so I'm going to try doing a recording. Hopefully that works better than what happened today in recitation. Okay, I'm going to share my screen and we're going to get started. Once again, like normal, if you have any questions for me, please feel free to email me. I have no problem answering questions. Whatever. All right. So we're going to go over the quiz 10. We're going to talk about chapter 17, 18 slides. We are going to go over a textbook problem, and we're going to go over 3 different problem-based learning questions. I know this is not ideal, but this is what we're going to do because this did not work. So, all right. So this is the schedule we have right now. So we are doing this recitation course asynchronously. We have 1, 2, 3 lectures until the next recitation, and then 2 more after for the exam. I believe the exam is on the 16th and not the 14th, but I'm not sure. So don't quote me on that. We'll talk about that next week. But yeah. All right, so the first quiz question. Which of the following statements about the cytoskeleton is false? The cytoskeleton is made up of 3 types of protein filaments. We know that's true. There's intermediate filaments, actin filaments, and microtubules. The cytoskeleton controls the location of organelles in eukaryotic cells. We talked about in class that the cytoskeleton can help control where the endoplasmic reticulum is in the cell. There was like a picture with 3 different fluorescent microscopy images where we saw that the microtubules lined up perfectly with the endoplasmic reticulum. So we know that's not true, or that is true. Covalent bonds between protein monomers hold together cytoskeletal filaments. That's not true. And then the cytoskeleton of a cell can change in response to the environment. We know that to be true. If you add different signaling molecules into the environment, the cell will change how it's expressing its microtubules or actin filaments or intermediate filaments. So C is the correct answer. Sorry, C is the correct answer. Number 2, phosphorylation of nuclear lamins regulates their assembly and disassembly during mitosis. If you remember, lamins are the molecules that are inside of the nucleus of the cell, and they're important during mitosis. So you add a drug to cells undergoing mitosis that inhibits the activity of an enzyme that dephosphorylates nuclear lamins. What do you predict will happen? Cells will arrest in mitosis with phosphorylated lamins. Cells will arrest in mitosis with dephosphorylated lamins. Cells will arrest in G2 with phosphorylated lamins, and cells will arrest in G2 with dephosphorylated lamins. Well, we know that the lamins are important in actually undergoing mitosis, and G2 is not happening in mitosis. It is part of interphase, so we can toss out C and D automatically. So then we have to remember which, is it going to be phosphorylated lamins or dephosphorylated lamins? Well, the answer is fortunately in the question. The enzyme that's being inhibited is the one that dephosphorylates these nuclear lamins. So we end up with just phosphorylated lamins. So A is the correct answer. I know that some of you were saying that you thought it was a trick question. It was not. All right, we're going to go over 4 different slides, and yeah. All right, so first thing I want to talk about is dynamic instability. Basically, what this means is that the microtubules can change whether they're growing or shrinking depending on the environment, and they are constantly doing both. On the plus end, the microtubules are attaching to a already formed tubule, and they grow, as you can see on the figure on the left. You can see here we have these monomers just adding, and it grows the microtubule. And then the GTP, this red dot on each of these monomers, will hydrolyze into GDP. And it will keep these filaments intact, but it means that they can disassemble. If you look at the picture on the right here, because there's no GTP holding these filaments, these monomers together, they'll easily just start peeling away, and breaking. The tubule will break apart. That's really the important thing to know here. And it needs for a microtubule to be stable, it needs to have a protein to protect both ends from depolymerization. So we need to have a protein that covers this end here and a protein that covers this end here. And we can see that here in figure D, where there's a protein on the right end, those pink ones, that protects the right end, and then the centromere down here on the left side protects the left end. And then there are still unstable microtubules elsewhere in the cell. You can see them, they have the arrows because they don't have a protected end on one end of the microtubule, so that they are not stable microtubules. All right, the next thing that I think is important to know is how actin and myosin filaments interact with each other and what that process looks like. So basically, we have the myosin head is attached to a binding site on the actin filament. ATP binds to the myosin thick filament. It hydrolyzes into ADP and inorganic phosphate. The phosphate jumps off of the myosin head, which allows the myosin head to bind back to the actin. Then the ADP pops off of the myosin head and pushes the actin filament forward, which is how we get that power stroke. And then we're back to where we started with the myosin head bound to a binding site on the actin filament. And we can repeat this process over and over and over again. And this is the main action happening in muscle contraction. All right. Okay, so then I wanted to talk about, there are 3 different checkpoints in the cell cycle, and there are 3 different tumor suppressors that are important to know. So we'll start with the checkpoints. So the first checkpoint we encounter is the environment favorable? Do we want the cell to undergo mitosis? If the environment is favorable, if the cell has the resources to divide, then we'll say, yeah, it is. And so we start entering the S phase where we make the new DNA. We go undergo DNA replication. Then we enter G2, which is another growth phase. And at the end of G2, we reach the second checkpoint, which is all the DNA replicated? That's kind of important if you want to have equal chromosome division to do different daughter cells. And then is all DNA damage repaired? We want to make sure that we don't have any extra mutations. We need to make sure that there's no mismatches. So we go through and we check all the DNA to make sure that all that's good. So if all that is good and it can all get fixed, we then go through mitosis. And we reach a checkpoint in mitosis, which is asking, are all the chromosomes attached to the kinetochores of the, are all the microtubules attached to kinetochores of the chromosomes so that we can easily pull them apart during anaphase and telophase? Okay, again, any questions, you reach out to me, that's fine. And then there are 3 tumor suppressors that are important to know. You saw them in the slides. I'm not going to go over the individual functions of each. If you want to know what they are, look at the slides, email me, that's fine. P53, I'm sure you've all heard before, is it's well known for regulating apoptosis. If there's a problem with P53, the cells will likely undergo apoptosis. Or sorry, if there's a problem with P53, it will not undergo apoptosis, causing a tumor. P21 is a CDK inhibitor. So if it inhibits CDKs, the cells cannot undergo mitosis. If 21 is bad, then it can cause the cells to divide anyway, causing a tumor. Same thing with retinoblastoma. If retinoblastoma has a problem, it will just start, the cell will undergo cell proliferation without having the CDKs to phosphorylate it. So again, another tumor suppressor. Yeah, again, if you have any questions, look at the slides, come ask me, whatever. Okay, yeah, and on to the next slide. There are the 5 steps of mitosis, which are prophase, which is when we start condensing the DNA into the chromosomes. Prometaphase, which is when we start seeing the chromosomes aligning toward the center of the cell and the nucleus has dissolved. Those are those lamins that I was talking about earlier. Then we go into metaphase, which is where we line up all the chromosomes along the center plate, and we have microtubules attaching to each of the kinetochores. Anaphase, which is when we have that checkpoint to make sure that all the microtubules are attached, and then we start pulling on the cell to make sure that to get it to start dividing. And then telophase and cytokinesis. Telophase is just the last step when we actually start to form the nucleus again and get all the DNA separated into the two cells, and the cytokinesis is just how we cut all the cytoplasm and all of the different organelles inside the cell into halves, so that both cells get a semi-equal number of each. Again, any questions, feel free to shout. But I'm sure you've seen all this before. Oops. All right. So then this is the book question I chose. Once again, not a super great book question, but yeah. So, which of the following changes takes place when a skeletal muscle contracts? Z discs move farther apart. That doesn't make a whole lot of sense. Because we're contracting a skeletal muscle, contracting means it's getting shorter. So the Z discs should get shorter and move closer together. Actin filaments contract. Actin doesn't contract. Myosin filaments contract. Myosin does not contract. So sarcomeres become shorter is the answer. Sarcomeres, if you remember, are the subunits of muscle cells. And when muscle contracts, sarcomere gets shorter. That is the answer. Contraction, shorter. That should have been, again, if you have any questions, feel free to reach out. All right, I'm going to go over these. So you are curious about the dynamic instability of microtubules and decide to join a lab that works on microtubule polymerization. The people in the lab help you grow some microtubules in a culture using conditions that allow you to watch individual microtubules under a microscope. You can see that the microtubule is growing and shrinking as you expect. The professor who runs the lab gets in a new piece of equipment, a fancy laser beam that can be used to sever the microtubules. She is very excited and wants to sever growing microtubules in the middle using the laser beam. Severing the microtubule with the laser will expose a new plus end on the shortened microtubule. Yay. Do you predict that the newly exposed microtubule plus end will grow or shrink? Well, we talked about earlier dynamic instability. As the microtubules grow, we start to see that GDP will stay attached to the subunits after GTP is hydrolyzed. And when there's just GDP, then we know that the microtubules will start to shrink because there's nothing, the GTP is not holding them together. So we would expect them to shrink if we sever the microtubules in the middle, because the GDP carrying subunits will start falling apart and peeling away, and the microtubule will shrink. All right, in a second experiment, you grow the microtubules in the presence of an analog GTP, which means that it cannot be hydrolyzed, and then you sever the microtubules. What do you think will happen in this case? Well, if we just have GTP, everything is going to stick together, and it will likely just keep growing. Again, it goes back to that dynamic instability slide that I showed. GTP, if it can't hydrolyze, GTP will hold this stuff together, the microtubule together. So with there still being GTP there, it will just keep growing like normal. Right, and then lastly, the drug colchicine inhibits microtubule polymerization and the drug Taxol stabilizes microtubules. Why do both inhibit mitosis? So colchicine inhibits microtubule polymerization if cells undergoing mitosis cannot grow their microtubules. They won't be able to bind to the kinetic cores to then pull them apart. And if you add Taxol to stabilize microtubules, then the microtubules will not be able to shrink, pulling those chromosomes apart. So both drugs inhibit mitosis. Feel free to take a picture of the answers here. Yeah. Cool. All right, number 4. No, number 2, whatever. Some lower vertebrates, such as fish and amphibians, can control their color by regulating specialized pigment called melanophores. These cells contain small pigmented organelles termed melanosomes that can be dispersed throughout the cell, making the cell darker, or aggregated in the center of the cell to make the cell lighter. You purify the melanosomes from melanophores that have either aggregated or dispersed melanosomes to find that aggregated melanosomes co-purify with dynein and dispersed melanosomes co-purify with kinesin. Given the set of data, propose a mechanism for aggregation and dispersal of melanosomes that accounts for the changes in skin tone. So I think it's important to know here that kinesin and dynein both move along microtubules. That's what that first GIF was at the very beginning of the presentation. That was a kinesin or a dynein moving along a microtubule carrying a vesicle. So we know that kinesin and dynein are on microtubules. So we have to know now which direction do they move. So if you remember from the slides, we would know that kinesin moves toward the plus end of the microtubule, which is out toward the outside of the cell, and dynein moves toward the minus end, which is toward the inside of the cell, which it makes sense. So the dynein would be associated with the aggregated melanosomes because the dynein would be pulling the melanosomes toward the center of the cell when they're aggregated, and the kinesin would be pulling the melanosomes toward the outside of the cell where they're dispersed. So that's the answer for that. They're on microtubules. Dynein moves toward the center of the cell, kinesin moves toward the outside of the cell. Cool. Last question. You are a cancer biologist working for Behemoth Pharmaceuticals, specializing in artificial sweeteners. Some chemists are coming up with 2 new compounds that taste sweeter than sugar and have 0 calories. However, when given to rats, they start forming tumors. You test the 2 new compounds against sugar for tumor promoting activity in cell cultures. You plate tumor cells in tissue culture plates and grow the cells in increasing concentrations of either compound 1, 2, or sugar. At the end of the 3 days, you count cells in each plate to determine the number of increase in cell number during the incubation period, which is presented below in the graph. So, do either of the compounds stimulate tumor growth more than sugar? Which one, and how do you interpret the data to come to your conclusion? All right. So once again, well, both. You can see that here. Clearly, sugar is the dark line. It makes very few tumor cells at almost any concentration. It starts to uptick at 10 to the negative 6, but both of the other 2 compounds start making tumor cells at much lower concentrations. So both make more tumors than sugar. What is the EC50 of compound 1 and compound 2? Go back to when we talk about 1/2 Vmax for enzyme kinetics, very, very similar. Go to about the 50% mark. So this goes up to about 1.1, 1.2. So then we go to like about the 0.6, 0.7 mark, and we see that it's about 10 to the negative 10 here for compound number 1. And then for compound number 2, once again, about 1.2, go to the 0.6 mark, it's about 10 to the negative 7. Very similar stuff. We remember we do this because it's at the linear point, so it's the most accurate. So we know what the kinetics are for these compounds. We talked about that in class a while ago. Okay. Your chemist colleagues tell you that both compounds are effective or they taste sweet with a concentration of 10 to the negative 9th. Which compound would you suggest they use to have as a sweetener to risk minimal tumor growth? So again, we look at this graph. At 10 to the negative 9, we see that compound 1 makes a lot of tumor cells, and we see that compound number 2 makes much fewer. So I would suggest using compound number 2 to make the sweetener, and also have the lowest risk of forming tumor cells. Pretty simple stuff. Again, if you have questions, feel free to reach out. Yeah, really sorry we couldn't get this to work in recitation, but hopefully this will account for it. I will go over these questions again if we feel like we need it at the end of next recitation. But yeah, I hope you all have a good weekend, and I will see you on Monday.